Hello and welcome to this Bio43 pre-lab video. In this video, we are going to continue talking about effective science writing, this time focusing on the discussion. Let's start by looking at the rubric. The interpretation category is closely linked to the discussion. Make sure your discussion covers every point under this category. And again, authority and structure/prose apply to the entire report, and will therefore be important for your discussion as well. First, you should interpret the results and explain why they matter. Remind the reader of your predictions and discuss whether or not the data matches them. You don't want to repeat specific data from the results, but you should reiterate the major comparisons between treatments and indicate if the differences are significant by restating the p-values. The major focus should be whether or not your hypothesis was supported. And once again, supported is the strongest language you can use. The results of a single experiment will not prove anything. Next, discuss any unexpected findings. There are many things this could include. Maybe the results did not match your predictions. Maybe the variation among replicates was unusually large. Maybe there are just a few odd data points. Maybe you made an interesting observation that is not part of your planned measurements, but is worth mentioning. Propose some explanations for these findings. Many students default to assuming that anything unexpected is due to error. And while addressing sources of error is important, you should also consider that everything went right and the unexpected result is real. These aren't canned experiments with known correct outcomes. This is the edge of knowledge. There will be things we can't predict. Propose a new hypothesis that could explain the unexpected result. The discussion is another part of the paper where you should cite outside sources. It's good to place your results in the context of past research. Do they mostly align with the results of previous experiments? Is there a contradiction? Can you think of a new hypothesis that would explain the conflict? Then what are the next steps you would take? Answers always lead to new questions. What experiment would you like to perform next to further explore this topic? You should talk about more than just repeating the experiment, but correcting certain errors. If everything had worked perfectly, what is the next experiment you would want to perform? Keep in mind that this is more than just hypothetical. Future Bio43 students will be continuing your projects, and may very well use the ideas you suggest here. You should conclude by discussing the broader significance and implications of your work. In your introduction, you should have started with a big picture. How have your results affected our understanding of that big picture? The overall structure of the discussion is essentially the opposite of the introduction. There you began with the big picture and ended with specific experimental predictions. Here, you begin by looking at how your results match your predictions, and expand to broader concepts, ending with the big picture. I hope you now feel prepared to write your discussion section. Again, remember that your writing guide and rubric are key resources you should be referencing while you write. See you next time!